Welcome back to our wet lab series. I'm Loa from Team IGEM Heidelberg, and today we're going to dive into the fascinating process of bacterial transformation. In the molecular biology, we often use DNA in the form of circular plasmids. Those plasmids can be assembled or edited using techniques such as Gibson assembly or Golden Gate assembly. If you're not familiar with those, you can check them out on our learning platform. Our plasmids carry a gene of interest which encodes for specific proteins which we need for further experiments. Our goal today is to insert those plasmids into bacteria, which then amplifies the DNA for us. This process is called bacterial transformation, where the bacteria takes up DNA from its surroundings. When the bacteria grows and divides, it also multiplies our gene of interest, creating more and more copies. Because taking up foreign DNA from the environment is quite stressful for a bacteria, we want to provide it with a good and nutrient-rich environment straight after, with the perfect conditions for a bacteria to grow on. That's why before we start, we prepare some plates with LB medium. LB is short for Luria bertani, and it's named after the two microbiologists that developed it. It has 4 ingredients. The first one is tryptone, which is a source of amino acids and peptides derived from the milk protein casein. It provides essential nitrogen and carbon sources for bacteria. We're going to take 10 grams of this and put it into our flask. Our second ingredient is yeast extract. This contains vitamins, minerals, and amino acids that are important for bacterial growth and metabolism. We're going to take 5 grams of this and put it into our flask as well. Our third ingredient is sodium chloride, which maintains osmotic balance and helps stabilize the environment for our bacterial cells. We're going to take 10 grams of this as well. Our fourth ingredient is water, but before we add that, we're gonna put in some agar, which is a gel-like substance derived from seaweed. And this will solidify our LB medium and later allow colony formation. We're gonna take 15 grams of this. And now we're gonna fill up this to 1000 milliliters of water. Now that we have all our ingredients, the next thing we want to do is to get the agar into solution. To do that, we're going to put the whole flask into the microwave and heat it until it boils. Let's go! First, we want to get all these bits into solution, so we're gonna shake it. So to make sure it doesn't boil over when you heat it up, we're gonna loose the screw top a little bit and then put it in. Now for a final step before we can pour the plates, we need to add one more ingredient to our LB agar, and that is ampicillin. This last step is to ensure that only the bacteria that has been successfully transformed will survive on the plate. Because our plasmids also contain an antibiotic resistance gene, the bacteria that will have taken up the plasmid won't be affected by the ampicillin at all. Now it's at approximately 60 degrees, a bit more, that's okay. We're gonna take 1 milliliter of ampicillin because we have 1 liter of LB agar and we want the ratio to be 1 to 1000. We will mix it. And now it's time to pour our plates. So we have this plate here. And now it's time to pour. And we'll make a thin layer of agar. Now it's time for the actual transformation. We first take our competent bacteria out of the freezer and quickly let it thaw. This bacteria has been treated with calcium chloride, which neutralizes the negative charge of both the DNA and the cell membrane. This reduces the repulsion between them, making it much easier for the DNA to interact with the cell membrane and eventually letting the plasmid enter the bacteria. Okay, so now we're going to pipette 1 microliter of a plasmid and introduce it to our competent bacteria. And to mix it, we're going to flick it like this with our fingers. And then we're going to put this on ice for 30 minutes. And now we're going to put them into a short heat shock. When the bacteria is exposed to these high temperatures, the small pores in its membrane open up, allowing the plasmid DNA to enter. And now we're going to place it back on ice for 5 minutes.
So earlier we prepared these LB agar plates with ampicillin added, so that we select for transformed bacteria that have the ampicillin resistance on the plasmid.
And now we are gonna plate out our bacteria on this LB plate. The recovery period is over, and we will now plate out our bacteria on this plate. We'll just put everything on here, and then we will evenly distribute it like this. And for the last step, it's very important to write down what you have on your plate. Today's date is the 20th September. My name is Loa Zink. And here we have LB with ampicillin. And the plasmid we use is called R8. So now our plate is ready. With our bacteria, we will turn it upside down so that if there's any condensed water, it will go here into the lid of the Petri dish, so it doesn't disrupt our bacterial growth here on the agar. And tomorrow we will see which bacteria successfully has been transformed or if we have any colonies.